Hello everyone, I'm Mr. Furlong, and today we're talking the cell cycle. The cell cycle is the life cycle of the cell, and it will go through several different stages. So when a new cell has just been formed, it is going to enter into growth 1 stage. So this is when the cell is doing whatever that cell is supposed to be doing. It's going to be in growth 1. Now, some cells will never go through the dividing process, and so they will actually go into what's called the G0 phase, and they will stay that way for the rest of their lives. But many cells at some point are going to divide. And when they do, a few things have to happen. First, there's going to be the S phase. So the S phase is the synthesis phase. Remember, to synthesize means to make or to build. So here we're going to be making a copy of all of the chromosomes. We're going to be making copies of things like mitochondria and other organelles. And once that is finished, it's going to enter into the G2 stage, which is the second growth phase. Here the cell is getting ready to divide, and then it enters into mitosis. So let's start off taking a look at exactly what's happening during mitosis. In mitosis, we end up producing 2 genetically identical daughter cells. Each has that full set of chromosomes. So we refer to these as diploid cells, di meaning 2, because they have 2 of every numbered chromosome. Now we use the abbreviation 2N. N, of course, can be any number. So for humans, N is 23. We have 23 pairs. 23 times 2, we have 46 chromosomes. What's happening during mitosis? Well, starting off in prophase, the nucleus actually breaks down. So that envelope disappears, and the spindle fibers are going to form. If we're talking about animals, these are going to come from the centrioles. Plants do not have any centrioles. This particular cell, this is an onion root tip, is undergoing prophase. So you can see the chromosomes, they have been stained. They're in no particular order. There's no nice definition from the nucleus compared to the rest of the cell because the nuclear envelope has been broken down. During metaphase, chromosomes will attach to the spindle fibers, and they will all line up in the center of the cell. So here we can see a cell that is in metaphase. All those chromosomes are right in the middle of the cell, very easy to identify. Now these chromosomes are going to start to split during anaphase. They move to opposite sides of the cell. Here we can see a cell that is undergoing anaphase now. One set of chromosomes going to one side, one set of chromosomes going to the other side. And then finally in telophase, the cells are going to physically divide and the nuclear envelope reforms. So here we can see a cell that is just beginning telophase. Then it enters into what is called interphase. Interphase is when the cell is doing whatever the cell is supposed to be doing. So in other words, it's in the G1 stage. We can also include the S and the G2 phase as part of interphase as well. Here we see a cell that is in interphase. You can clearly see a distinct border around the nucleus. We don't see any specific chromosomes because the DNA is now in its chromatin shape, and the cell is doing whatever this onion cell is supposed to be doing. Now, in order to control the cell cycle, there are a variety of checkpoints. So at the end of the G1 stage, we have what's called the G1 checkpoint. This is the main checkpoint for all cells before they get ready to divide. And what it's looking for is, has the DNA been damaged? Is this something that can be replaced? If so, let's replace it. It can check to make sure that, is there enough room for there to be more cells? If there's already too many cells there, we don't want to make more cells. And it's also going to make sure, is there plenty of oxygen? Is there plenty of nutrients available for these new cells? If any of these things occur, the cell should not divide, and it would actually go into what's called apoptosis, which is cell death. The second checkpoint is the G2 checkpoint. Here it's looking to see that the DNA has been properly replicated. Remember, during the S phase, this is when DNA replication is going to take place. It's going to look for any errors in the DNA. If the DNA has not been replicated properly, if there are too many errors in the DNA, the cell should destroy itself. It should go through apoptosis. Lastly, we have the M checkpoint. This is looking to make sure that the chromosomes are actually attached to the spindle fibers properly. We want to make sure that the replicated chromosomes separate so that one goes into one cell, one goes into the other cell. If not, you guessed it, apoptosis is going to take place. So what happens if these checkpoints are not met and the cell continues through the cell cycle anyways? What develops is something called cancer. Any type of disruption to the cell cycle will result in some type of a cancer, or it should go into apoptosis. It should go into cell death if it doesn't. As of spring of 2013, there's been 140 different cancer genes that have been identified. More are always being found. It's estimated that there's probably going to be somewhere less than 200 genes. So we may have found most of all of the genes that potentially could cause cancer. So what normally happens is you have 2 copies of every single one of these genes. Now these genes are supposed to, again, make the cell go through the cell cycle so that we can go through mitosis, make more cells. But we have other genes that, again, they're checking to make sure that things are stopped. So think about this. If we have one mutation and this gene is telling the cell, divide, 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 well, we still have these tumor suppressor genes that are working that are like saying, stop, 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 and nothing happens. Those cells would just go through apoptosis. If we also have a mutation in a tumor suppressor gene that's supposed to put the brakes on, well, we still have one gene that's making the proper protein that's saying, nope, stop, we're not making any more cells. The cell will go through apoptosis. One of the problems is, what if both of those genes have a mutation in there? And now we have a gene that is putting on the gas saying, make more cells, and we have no brakes. And so more and more cells are made, developing a tumor, hence we end up with a cancerous tumor. As we are finding more and more about these different types of mutations, that leads to better treatment options for us. So that is the cell cycle and mitosis, and I'll see you in class.